Okay, hello everyone. I'm starting with few announcements. Please keep your camera active during the seminar. So regarding the communications, it is a student responsibility to check the McGill email regularly for any communication. Do not use your personal email when you send us messages and include your McGill ID number because it makes our life much easier. So, as you know, I sent an email recently regarding the emails that you can use for communication with us. PhD, it's Sharon, and you have her email. The non-thesis programs is Misha, and also you have her email. And the MSc thesis and harmonized stipend, so everything that you receive from the Jeff funds, it will be me. So please don't send emails everywhere, or if you don't see that we didn't answer, please give 48 hours until we are able to respond. For the research day communication, please send us the email, use the email SIS. Do not use the the admission email in any situation. You are already in the program. It's very hard for us to forward emails everywhere. So please, this one, I know you use it before you are admitted in the program. Don't use it anymore. Progress tracking, it's a reminder to have your RAC meetings in the first 3 months since you started the program. Complete the tracking forms, upload them on your progress. Inform us that you uploaded because we can't guess that you already did, and we will process your milestones. Again, a reminder, we are open for fast tracks for the fall 2025. If you have a CGPA of 3.5 or higher and you're interested in pursuing a PhD, please let us know and we can help you with the fast tracking program. We have supplemental financial assistance, a minimum $2,000 for the first 5 eligible fast track students. We want to thank the Surgical and Interventional Science Graduate Student Society for helping us to organize this virtual seminar, especially to organize the discussions with the students and presentation with the students. Thank you, Pega, for helping me find the students who's going to present in our seminar. And today's moderator for the students will be Tarek. Do not forget, we have presentation awards, first prize, second and third prize, as mentioned on the screen. Special mention, $100 for everybody who participate. And of course, we have an additional seminar question award of $50 for the first two who put more than 5 questions during the seminar. If you are interested to do a summer medical internship, the date of registration will be at the end of March. Please send us a CV by the end of March to see if you are selected by any of our companies. And if you are selected, you'll receive permission to register. The courses are 6 credits per course. So yeah, if you are interested to do something different, let us know. Okay, thank you for sending your abstracts to the research day. Um, Perfect. You will receive some emails from me regarding the formatting or anything. Now, do not forget that we have a limited number of posters this year. So we'll have 15 oral presentation and just 30 posters. So not everybody who sent will be accepted to present. And please do not forget to register at the link on our website and here on the screen. Thank you very much.
Thank you very much, Laura. Good afternoon, everyone. So we are honored to have with us Professor Christine Lemaitre, who is an esteemed expert in musculoskeletal cell biology and tissue regeneration. She's based at the University of Sheffield's Medical School, where she co-leads the osteoarthritis and disc research group in the Division of Clinical Medicine. Professor Lemaitre's research focuses on understanding the pathogenesis of musculoskeletal disorders, particularly intervertebral disc degeneration and also osteoarthritis. Her work has been instrumental in developing novel regenerative therapies to tackle these conditions at their root cause, rather than just managing the symptoms. With over 120 publications, 2 patents, and more than 25 million pounds in research funding, her contributions have significantly shaped the field. Her impact is further reflected in her H index of 50 and over 10,600 citations. Beyond her scientific achievement, she is also a dedicated mentor, having supervised more than 25 PhD students to completion, with 7 currently under her guidance. Today, Professor Lemaitre will present on understanding the pathogenesis of intervertebral disc degeneration and osteoarthritis to develop new therapeutic approaches. So this is an exciting opportunity to gain insight into cutting-edge research and potential future treatments for these debilitating conditions. Without further ado, please join me in welcoming Professor Christine Lemaitre.
Thank you, Faxon. So it's an absolute delight to be invited to talk to you all today, and hopefully this will spark some interest in a number of the students staying in musculoskeletal or moving across to musculoskeletal, because it really is the best topic to research. I've been researching it for too many years now. So thank you very much for the invite. Just to say, so in terms of potential conflicts of interest, I am an inventor on patents for hydrogel systems, which I include in the presentation. And I act as a consultant for a number of companies and receive funding from some of these companies as well. So I'm going to start off with low back pain and talk about kind of disc degeneration first of all, and then I'll come to osteoarthritis towards the end. A lot of the pathogenesis of disc degeneration and osteoarthritis are common, and there's a lot of similarities. So actually looking at both of these conditions together, we can actually try and develop new therapies for both conditions kind of simultaneously and actually target a much larger patient population. So why low back pain? So low back pain is the biggest cause of disability worldwide and has been ever since they started measuring kind of the disability, the global burden of disability back in 1990, and low back pain has been the leading cause of years lived disability since then. It affects about 600 million people each year, and 80% of you, I'm afraid, are going to get low back pain at some point in your lives. But it doesn't just affect people physically, it also affects you psychologically and also socially, affecting actual normal daily living and preventing a lot of activities and even sleep. And what's really shocking is that 85% of chronic cases of back pain are diagnosed with no specific cause. And this is mainly because for these individuals, there's no treatment options. So often medical practitioners just fail to bother trying to diagnose it because actually there's not actually treatment options out there. So it has a really high economic burden, and we really do need to find better treatments rather than just giving people painkillers, for example. So one of the big causes of back pain, and this is associated with about 40% of all chronic back pain cases, is disc degeneration. So the discs are in your spine between your vertebral bodies. I'm going to put a little laser pointer on. So if you've got, so these are your vertebrae, and then in between them are your intervertebral discs. And you can see that these 2 discs at the bottom appear black on this MRI image, and that's because they are degenerate and they've lost a lot of the water content, so they appear darker on the MRI. Now, disc degeneration does increase turn the laser off, with age, but actually it's in individuals who are kind of 55 years and under, who are the main working class individuals, where the association between disc degeneration and back pain is strongest. So it's these individuals that we really need to try and target and treat the causes of disc degeneration to prevent the chronic back pain. So what occurs during disc degeneration? So if we look at the discs in a bit more detail, the top disc here is a normal disc. And you can see that you've got this central region here, the laser point on again, sorry. The central region here is known as the nucleus pulposus, and this contains lots of proteoglycans, which holds on to lots of water. And that basically is a hydrogel. And this is constrained around the outside by these concentric layers of collagen known as the annulus fibrosus. And I often use the analogy of a jam donut. I don't know if you have jam donuts in Canada, but the dough on the outside is very much like the annulus fibrosus, and it holds in the jammy middle within the donut. And if you hit the donut hard enough, you'll squeeze that jam out. And that's basically what happens when you have a herniated disc, is the jammy middle will squeeze out of the dough and cause pressing of the nerves. Now, during degeneration, the cells within the disc change their phenotype and they start to produce matrix degrading enzymes, which will degrade the matrix. And also you get this switch in the normal proteoglycans and collagen type 2 to more collagen type 1 and smaller, less aggregating proteoglycans. This means that the disc becomes much more fibrotic until eventually in the severe degeneration, you can actually get fusion as well within the discs. So in the low to moderate grades of degeneration, this is where we want to try and target regeneration, which I'll talk about in a little bit. And then in the severe degeneration, it might be that we just need to try and hurry up the fusion process to prevent some of the painful stimulation. So the intervertebral disc is actually the largest avascular tissue within your body, and the blood vessels are only found on the very surface of the annulus fibrosus and also the edge of the cartilaginous end plate, which is the top region separating the disc to the vertebral bodies. And this means all of the nutrients within the disc have to come through this thick extracellular matrix that the disc is made up on, meaning that the disc has low oxygen and low nutrient supplies, and the cells have to survive within this harsh environment. Now, during degeneration, you do get fissures that form within the annulus fibrosus, this outer region here, and you can get nerves and blood vessels ingrowing. And also, this degeneration is often associated with vertebral bone marrow lesions known as modic change. Originally, these modic change lesions were thought to be due to altered mechanical stress, because as the disc degrades, this then passes the mechanical stresses differently onto the vertebral bodies. But actually, it's now known that there's potentially two etiologies. One of these is once you have the cracks and fissures within the end plate, you get leakage of different agents from the disc, such as cytokines or broken down extracellular matrix that leaks into the vertebral bone marrow, causing activation of immune cells. You can also have a potential infectious etiology where the presence of bacteria, potentially from the disc itself, can invade into the vertebral bone and again cause this osteolysis effect and the bone marrow lesions. But the question whether there's bacteria present within the disc is quite a controversial area. So a lot of the studies which were originally done looking at the role of bacteria present within the disc were on herniated material. So this is material which is squeezed out of the disc tissue and it's now outside of the disc, so it's exposed to the surrounding area. And it may be that the bacteria that's been found is just been there because it's herniated. And a lot of the studies have also used kind of candidate approaches where they're looking for individual bacteria, and actually C. acnes, or it used to be P. acnes, is the predominant bacteria that's been investigated. Most of the studies to date have used either attempted microbiological culture, which has the big disadvantage that only bacteria that grow in the environment you're trying to grow them in will grow. So actually, remember, the disc is low oxygen, low nutrients. So if you take them out of the disc and then put them in a high oxygen environment with lots of nutrients, they're just not going to grow, so you won't identify them. And then more recently, there's a technique called 16S RNA sequencing, which is where you can actually sequence specifically the bacterial DNA to look whether there's bacteria and work out what bacteria are present. But one of the main issues with 16S RNA sequencing is this is really easy to contaminate. So it could just be that the bacteria you're identifying is from contamination from the sample, from the outside, during surgery, from the skin. There's lots of different sources. But there has been a few studies which have looked at the 16S sequencing, and these have shown, this is a systematic review, which has shown that in studies which have demonstrated potential presence of bacteria within the disc, there's an increased risk, shown on this risk index, of modic change, and also an increased risk within males as well. So what is the association between bacteria and modic change? And this work by Stefan Dudley, who's in Switzerland, demonstrated that if you take discs and count how many copies of this particular bacteria, C. acnes, is present, then you can see that even in control discs shown on the top here, that actually C. acnes was detected within these control discs. But if you look at the discs which are associated with modic change vertebrae, you still have these lower regions in some of the discs, but in some discs you have these high copy numbers of C. acnes. And more recently, with the 16S sequencing data, it's been shown by 2 studies, one by Rajesh Sakaran in India and one by Stefan Dudley in Switzerland, that again, non-degenerate discs seem to have bacteria in, so you've still got them present within the controls, shown in green here, and then non-modic change discs over here. But actually you get this apparent dysbiosis, so there's different bacteria present once the discs are either degenerating or they're associated with different modic change types. So what we wanted to look at, though, is this bacteria truly within the disc? Because, as I say, a lot of these studies have either taken bacteria and tried to culture them, which has major issues, or using the 16S sequencing, which then the argument is always, well, you just contaminated it and it's not true. So using a technique known as immunohistochemistry, where you can stain specific bacteria, we've looked within 95 discs, and these are all discs which are not herniated. So we get samples and the surgeons tell us or not whether the annulus is still intact, and only the ones which are still intact are the ones that we've utilised within our study. So we've looked at 2 key bacteria, which are the ones which are identified in the most numbers of studies, C. acnes and Staph aureus. And what you can see is that the C. acnes and the Staph aureus are indeed present right in the middle of the disc. But what's really interesting is they're internalised in the disc cells. So we don't see biofilms of the bacteria within the matrix. They're all actually inside the disc cells themselves. And if we go through and count how many disc cells contain these bacteria, you can see in these graphs, so purple is in females and the green is in males. And really interestingly, with Staph aureus, that's mainly found within female discs. So we don't see very many males with Staph aureus within their discs. Whereas C. acnes was found across every single sample we looked at, although there was different numbers of cells containing the C. acnes, with an increase in the numbers with age, although it wasn't significant, and there was some changes potentially with degeneration and modic change, but again, no significant changes there. So what's the potential kind of effects of these bacteria, and do the levels of the bacteria correlate with some of the catabolic factors that are associated with disc degeneration? So here again, using immunohistochemistry, we've looked at the levels of key cytokine, interleukin-1, which is upregulated in disc degeneration, and then degradation enzymes, so MMP3 and ADAMTS4 are 2 key degradation enzymes, and these are all increased in degeneration. Gastrodermin D is a factor that's associated with pyroptosis, which is a form of cell death that's associated with bacteria. So again, we were looking whether there was an increase in Gastrodermin D when you see increases in the bacteria. So looking at the correlation matrix shown in the table, you can see that we do have a correlation of C. acnes with age and also with MMP3 and Gastrodermin D, and nearly significant with interleukin-1, so it's 0.069. So if we add more data sets, this may become significant. But Staph aureus shows no correlation whatsoever with any of these catabolic factors. So how do cells actually recognize these bacteria? So within immune cells, there's lots of different potential receptors, and this is a very busy slide, but just to kind of highlight, there's key receptors on the cell surface known as toll-like receptors, and then also intracellular receptors such as NOD2 and NLRP3, which will recognize bacteria within immune cells. And it's known that within disc cells that discs express toll-like receptors. And in fact, this is some of the work of Lisbeth and Derek, which has shown that if you treat disc cells with toll-like receptor agonists, you can promote catabolic factors, so cytokines and nerve growth factors. But what's not known is disc cells express NOD2, because this has only been shown to date within immune cells and epithelial cells. So again, using immunohistochemistry, we've looked at whether the disc cells express these different factors. We already knew they should express TLR2 and TLR4, and we also see these in our disc samples. But we also see that NOD2 is highly expressed as well within the disc. And if you look at the number of cells expressing NOD2 and the number of cells containing C. acnes, we see positive correlation between C. acnes and NOD2, and then also positive correlations between the different receptors as well. So what can the C. acnes actually be doing within the disc? So it's known from previous studies, both with in vitro work, if you treat disc cells with C. acnes, or P. acnes it used to be called, or inject it into animals, you can induce features of degeneration. But a lot of these studies have used really high concentrations of bacteria when they've injected it into the discs or done co-culture studies. And because within our human disc samples, we didn't see any evidence of biofilms, which is what you would expect to see if you had lots of bacteria present, we only ever saw it internalize into disc cells. We wanted to look at what effect could lower doses of this bacteria have on disc cells. So we've looked at both the co-culture with the bacteria themselves, and then also treatment with peptidoglycan, which is the cell wall component of gram-positive bacteria. So if we co-treat with, this is actually Staph aureus here, labelled with green, you can see the disc cells can internalise them. And if we look at a whole host of different cytokines and nerve growth factor and vascular endothelial factor, you can see that when you treat with peptidoglycan, you get promotion of a lot of these secretory factors. But when you treat with Staph aureus and C. acnes, you don't see the same increases. And if we look at intracellular signaling pathways, which are activated in kind of catabolic processes, again, peptidoglycan causes an increase, but the lower doses of Staph aureus and C. acnes don't. And in fact, if we look at some of the secreted factors in a bit more detail, you actually see that when you treat with kind of Staph aureus or C. acnes, it actually causes a decrease in the levels of some of these cytokines and vascular endothelial growth factor, suggesting potentially that the bacteria, or at least certain species and strains, could actually be protective to disc cells rather than inducing catabolism. Interestingly, if you look in other species, so this is in bovine discs, bacteria are actually present within bovine discs as well, so it's not just during degeneration. You can see that the C. acnes across multiple regions, as is Staph aureus. So the story around bacteria isn't understood fully, and we do need to do a lot more work understanding exactly what types of bacteria are present, what strains, and what they are actually doing within the disc. So I'm going to switch tact a little bit now onto some of the other kind of cellular processes that occur during disc degeneration. The first of these is looking at cellular senescence. So cellular senescence is when cells basically age, so they lose the ability to keep dividing. And we know that these accumulate within intervertebral disc cells, which are degenerate. So I showed many years ago, back in 2007, I think it was, where we actually see that you get an increase in senescent cells within degenerate discs. And if you take cells out of age-matched non-degenerate and age-matched degenerate discs and culture them in the laboratory, those from the non-degenerate discs have a much greater capacity for division than those of degenerate, showing that the degenerate ones are more senescent. And also, if we look at telomere length, which is an indication of replicative senescence, you can see that the ones from the degenerate disc, shown in blue, are much lower as soon as you take them out of the disc already, whereas the non-degenerate ones just decrease with increase in proliferation, as you would expect normally. And what's really interesting is these senescent cells also show increases in catabolic enzymes, so things like MMP13, and this is now known as the senescence-associated secretory phenotype, and it's well known now that senescent cells will actually increase lots of different factors within the disc cells. So what regulates the release of these senescence-associated secretory factors is activation of a pathway known as the cGAS-STING pathway. So if this is a cell, and this is the outer membrane, so I'll put my little laser pointer in as well. So this is the outer membrane, and then this is the nucleus membrane with your DNA in its right place in the nucleus. If the DNA becomes damaged, this DNA gets recognized by a protein, gamma H2AX, which we've shown is upregulated in degenerate discs. And it then shuttles the DNA out of the nucleus so it can't get transcribed. And again, we see cytoplasmic DNA within degenerate discs. And this is then recognized by a protein called cGAS, which is also increased in degenerate discs. And this causes activation of the STING pathway, which causes activation of all of the secretory factors. And interestingly, we've shown that the levels of cGAS correlate really nicely with the levels of senescence, shown here with P16 INK4A levels. And in females, there's actually slightly higher levels of cGAS than there is in males as well. So again, some gender differences here, which need to be investigated further. So what's actually inducing the senescence within the disc isn't fully understood, and it's probably a bit of a mixture. So senescence can either be induced by what's called replicative senescence, which is caused by every time a cell divides, your telomeres, which protect the DNA, at the ends of your DNA, they get a little bit shorter, and that's actually there as a protective mechanism. But when the DNA gets to what's called a critical limit, the cells can't divide anymore, and the cells become senescent. But as well as replicative senescence, you can also get a form of senescence known as stress-induced senescence, where cytokines such as interleukin-1, which we know is upregulated in the disc, or reactive oxygen species, again increased in the disc, can drive this senescence. So there's a number of studies which are ongoing at the moment trying to target cellular aging. And one of the ways that you can do this is by using senolytics to try and kill off the senescent cells. And this has been studied in a number of studies, including a number from Montreal, so from Lisbeth Haugland's lab, looking at various different senolytics to try and clear these senescent cells out of the disc. So within a recent Versus Arthritis grant, we're also doing a comparison of some of these senolytics against some small molecules which can inhibit specifically the cGAS STING pathway. And the idea is can we actually target inhibition of the kind of detrimental effects of the senescent cells by inhibiting the cGAS STING pathway, so you're preventing kind of the increase in these inflammatory cytokines. So this is work that's only just started really, and this is just some initial data that we've got where we can culture pieces of human disc tissue within these little silicon rings, which prevents them from swelling, and we can maintain their normal phenotype. And if we've treated these human disc tissue samples with various different senolytics, and then this is just the initial measure looking at the metabolic activity, you can see with quercetin, which is one of the senolytics, we do see a decrease in the metabolic activity with our disc tissues, and we're now kind of sectioning through to see whether this is because we have killed off senescent cells. But interestingly, two of the other senolytic agents have less effect. So it's also whether we do need to combine them, like Lisbeth's demonstrated. She uses the RG7112 and the Ovanel in particular. So we're now looking at the STING inhibitors as well. So another kind of key factor within disc degeneration that could be a good target for degeneration is the small molecule cytokine interleukin-1. So this was actually work back from my PhD, where I showed that IL-1 was increased during degeneration along with its receptor, IL-1 receptor, and this drives lots of the changes that you see during disc degeneration. So it can release neurotrophic factors and vascular endothelial factors, driving the nerve and blood vessel ingrowth. It can alter the matrix that's being produced and cause a switch from the normal matrix to abnormal matrix. It can drive a whole host of degradation enzymes and other cytokines and chemokines. And as I said before, could also be a potential inducer of cellular senescence. So one of the questions was often, well, is the increases in IL-1 that you see during degeneration, is this cause or effect? So a number of years ago, we looked within a knockout mouse model where you can remove the natural inhibitor of interleukin-1, known as interleukin-1 receptor antagonist. And you can see when you knock this out, you get spontaneous degeneration. So you get this spontaneous increase in disc degeneration, which is associated with increases in cytokines and matrix degrading enzymes, and an increase in cellular senescence. So IL-1 can definitely drive a lot of that pathogenesis within disc degeneration. So more recently, we've been working with an American company, Pacira, who have developed an IL-1RA gene therapy approach, which is actually in clinical trials for osteoarthritis, to see whether we can utilize this within disc degeneration. So in the degenerate explant culture system, which I showed before, again, we can cause an increase in the levels of IL-1RA via our gene transfer, and also an increase in aggrecan, and importantly, an inhibition of lots of the cytokines and degradation enzymes and the catabolic factors. So this is looking really promising, and it could be a potential new treatment. But that's all about kind of halting degeneration. How do we actually regenerate the disc? So in order to regenerate the disc, one of the crucial kind of functions of the disc is it's a mechanical structure. It undergoes loading and needs to withstand the normal mechanical loads that you experience during normal daily life. So we need to restore that mechanical properties of the disc. We need to consider the degenerate niche. Do we need to inhibit it? Do we need to replace dysfunctional cells? And do we need to inhibit process of degeneration? So something that we've developed is an injectable hydrogel system. So this is temperature responsive. So it's liquid above body temperature, and you can inject it through very, very fine needles so we don't damage the annulus fibrosus. And on the right-hand side here, you can see that we've put a red dye in so you can visualize it. And there you can see that it fills. Sorry, I restarted the video then. Yeah, let's fast forward. Sorry. And then rewind. There we go. Oh, never mind. So you can see that it filled the actual cracks and fissures, and then it's gelled instantaneously. If we look at this within a whole cadaveric disc, and this is actually something I did when I came up to Montreal pre-COVID, came over for a week and actually spent some time in Elizabeth's lab, utilising the cadaveric organ samples. And here you can see we've injected it into the disc. We've added a radiopaque substance, so you can see it's filled the cracks and fissures within the actual disc. And if we look at the MRI images, so pre-injection, you can just about make out the cracks and fissures. Oh, sorry. Pointer. You can just about make out the cracks and fissures within the actual disc here. And then when we've injected it, the gel is rich in water, so it appears white on the MRI, and you can see hopefully it's filled all of those cracks and fissures. And what we saw is following the injection, you get this immediate increase in disc height, shown in the blue bars, and also an immediate increase in the actual kind of mechanical strength of the Young's modulus of the discs. And of course, the last thing you'd want to happen is when that individual gets back up after surgery, you don't want the disc to herniate or the actual gel to squeeze back out. So here we're loading discs to failure, and this bottom one here is about to fail. You'll be able to see a herniation happen in a minute where that arrow is. It's about to pop out. There it is. So there it's squeezing out, but that's actually the central disc region, the nucleus pulposus, not the gel itself. And all the discs failed at 2 to 4 times the maximal physiological load, which gives great confidence that actually the gel will stay in. But this loading is pretty simple. It's just a simple compression. So what we've also now done is applied much more physiological loads, such as those mimicking tying your shoes or sweeping the floor. And you can see this is much higher forces that are being applied to the disc tissues. And even under these loads, we still see that the gel is retained within the disc and there's no herniation. We also see that within these discs, if you look at the amount that the disc moves about, so its range of motion, when you induce degeneration, you get this increase in range of motion in all different axes, so in flexion and extension and lateral bending, and in axial rotation, you get this increase in the range of motion. But if you inject our gel in, you then see this decrease in range of motion, demonstrating it's starting to stabilize the spine. And again, we see an increase in disc height as well. So that's kind of gel alone. We could also potentially add cells in as well. And there's lots of different cell sources we can utilize. If you want to read more, I'd encourage you to have a look at this excellent review that my PhD student did. And one of the key cell sources is stromal cells. So these you can access either from bone marrow or from fat tissue. So we wanted to look at, if you mix these cells with our gel, can we drive those cells to differentiate in towards disc cells? And sure enough, if you mix bone marrow progenitor cells with our gel, it induces differentiation. So they turn into disc-like cells with increases in aggrecan, COL2, and chondroitin sulfate. But then we wanted to say, well, do we need to use bone marrow derived, or can we use fat tissue? Then effectively a patient could have liposuction, for example, and then be treated as well. So you could treat the back with their fat effectively. So here we're looking in two different gel systems, an MP gel, which is for MP tissue regeneration, and B gel, which is for bone. So looking at the MP markers, PAX1, aggrecan, and COL2, you see that both bone marrow in red and adipose derived in black both differentiate just as well. So you get this increase in all of the MP markers in the MP gel. If we look at a bone marker, osteopontin, this is increased within the B gel, but again, there's no difference between the bone marrow derived and the fat derived cells. So this could be a potential source for new therapeutics. But what about other potential cells? So within the development of the disc, one of the key kind of juvenile cells is called the notochordal cell. And within humans, we lose these notochordal cells during early stages of development. And actually all species which lose these notochordal cells induce degeneration. So species that retain them don't get degenerate discs. So we wanted to see if we take notochordal cells from pig discs, which keep them, and put these into our gel system, can we keep their phenotype? And indeed we can. So if you want to read more, do have a look at that paper. But accessing pig cells for therapy is not really going to be very good for kind of treating people that don't really want pig cells injected in. So we could also use induced pluripotent stem cells. So here we've looked at inducing MP cell differentiation with induced pluripotent stem cells. And here you can see that, again, we're inducing this differentiation with our gel system. If we look, remember the disc, though, is rich of cytokines, so interleukin-1. So what happens if we put cells into this environment is it could actually increase the catabolism. But what we've shown is if you put cells into our gel, they actually decrease sensitivity to IL-1. So you get this knockoff of the receptor expression by cells within our gels, protecting them effectively from that catabolic environment. And indeed, if we inject our gel into disc tissue samples, either alone or with cells, you get this kind of promotion of aggrecan and COL2, so the anabolic factors, and you start to inhibit a lot of the catabolic factors, so IL-1 and the matrix degrading enzymes. But this is all in kind of unloaded situations. What happens when you apply load? So first of all, we looked within a goat organ culture model, and this was in Amsterdam, where you can take goat discs and culture them in the bioreactor. We can induce degeneration and then treat them with our gel alone or gel with cells. And you see that actually both induce decrease degeneration. And if you look at the actual histology, you can see within the black lines here, this is where the gel is. And if we zoom in, we can see cells from the injected gel without cells, the native cells are starting to migrate into the gel and actually start producing matrix. And we see again an increase in aggrecan and COL2 when we've injected our gel in. And what was fascinating was, again, we get this total inhibition of all of the catabolic factors, so matrix degrading enzymes and cytokines. But this is in a goat. So together with Lisbeth, we looked within the human organ culture model, where we can actually culture the living human tissues within a bioreactor system. And just to cut a long story short, after 4 weeks of culture, we switch from human discs with cracks and fissures in to filling in those voids and actually starting to regenerate the tissue and totally inhibiting all of those catabolic factors. So I'm just going to quickly go through some of our most recent in vivo safety data, and then I'll talk a little bit about osteoarthritis. So here we're inducing degeneration in a sheep model, and this is really to look at the safety of the gel injected into the sheep. So here they induced degeneration with a nucleectomy, which is where you take out a bit of the disc and it induces degeneration, leave them for a month and then treat them. And after 3 months, All of the sheep were very happy. There was no issues, no risk of any in vivo safety. And if we look at the peripheral blood and immunological activation, we saw no evidence of activation of immune cells following the treatment. There was some activation of immune cells following the degeneration, which is an injury response, but there was no further activation when we injected our gel or cells in. And one of the ways we can look whether the cells we've injected in are still there is by looking at this specific DNA sequence that humans have but sheep don't, known as allo sequences. So here we've actually taken all the different organs from the sheep to look at whether or not those cells which we've injected in are moving into different tissues. And basically none of the other tissues had any allo sequences in, so there was no migration of cells into other tissues. But if we take the discs, we still see allo sequences after the 3 months in the sheep that is injected with these cells, demonstrating they're at least surviving for 3 months. And then looking at some of the histology, you can see that when we've actually injected our gel, you can just about see this mottled appearance. Put my laser back on. So this mottled appearance here, that's where the gel is. And then when we've injected it with cells, hopefully you can see these little blue blobs, there is an increase in cell density within the disc that we've injected with cells. So we're currently going through all of the kind of immunomarkers to see exactly what's going on within these discs to understand this greater. This gel system has now been licensed to a company, Orthoson, and they've been doing work where they're actually moving now to do cold injection. And this was some of their MRI images of cadaveric spines where they've injected in the gel. And you can see again, the white is where the gel is, and it's filled all the cracks and the fissures within the actual disc tissue. And this is now progressing to kind of large animal studies for hopefully regulatory approval. So hopefully from kind of initial discovery and development in the lab, hopefully in a couple of years, we'll be able to be in clinical trials for this therapy. So I've just got a little bit that I'm going to touch on osteoarthritis, really to demonstrate the similarities between OA and disc degeneration, and to highlight some of the culture systems that we're doing where we're trying to keep the different joints of the together in culture. So with osteoarthritis, this is a disease of your kind of the rest of the joints of the body, particularly the knee and the hip are most affected. So this is obviously a knee. And it's not just the cartilage that gets affected. So you have defects in the cartilage, the bone, and also the synovium. So all of these tissues are affected. So when we're trying to understand osteoarthritis, we really need to look at all of these tissues together. And what's interesting is during osteoarthritis, similar that we saw within disc degeneration, you get these cracks and fissures forming within the subchondral bone. So actually the bone tissue becomes more exposed to the cartilage. The bone can also release factors into the synovial joint that the synovium will also respond to. So the 3 tissues really have this increased crosstalk. So what we're trying to do is develop culture systems where we can maintain these 3 systems together. So these types of cultures have been around for a number of years, which are kind of where you take plugs of the osteochondral explant, which is kind of the layer of cartilage on the surface here together with the underlying bone, and you can culture this within the laboratory. But the cultures today have mainly focused on cartilage responses. So you can see in these images over here, the bone, which is underlying, is actually dying. So they're not maintaining the viability within the bone regions, and it's just the cartilage that can be used and looked at. So what we've been able to develop is a way of keeping alive the cartilage and the bone all together. So we get kind of osteochondral tissues from knee replacement or hip replacement. We can take these little cores, and similar to when you culture in kind of a disc, you flush out all of the bone marrow from the bone region so that your actual nutrients can get into the bone. We then dip the cartilage into agarose, which is sterile, and that prevents the proteoglycans from leaching out into the media. And then we culture them free floating within culture media, which is low glucose and low oxygen to mimic the environment of the actual joint. And now we're using these to kind of understand better pathogenesis, screen new therapies, or also applying mechanical load. And I'm going to just jump through quickly some of the data on these, but basically all the tissues stay alive. So we've got living bone, living cartilage. The matrix looks exactly the same as it should do from previous cultures. And you can see you've got chondrocytes expressing COL2 and the osteoblasts expressing ALK-Phos, which is exactly what they should be doing. But what about the synovium? So we also now have developed this co-culture system where we can culture little pieces of synovium together with the osteochondral explants. And this is just taken before we put the media in, just so you can visualize it better. So you can now get this crosstalk between all of the 3 different tissues. And what we've demonstrated that is we can culture these for up to 3 weeks. And this is actually after 3 weeks of culture. Within the low-grade tissues or the high-grade tissues, these look pretty much the same as they do when you first take them out of the body. And with the synovium, you have all of these different regions which represent the different types of tissues that you get within synovial tissue. And these are all maintained within the culture system. So if we look at collagen, for example, with Mason trichrome staining, you can see that you've got really good retention of the collagen within the different tissues, potential little bit of an increase, which is probably because you've got higher nutrients, but mainly the same. We don't get any increase in apoptosis. So this is looking at caspase 3, which is an apoptotic marker, and really low levels of apoptosis present. Usually if we quantify it, you can see there's less than 20% in any of the things. IF is immediate fixation, so you can see that there's no increase in cell death during the culture. And if we look at some of the markers, so type 2 collagen should be there within particularly the chondrocytes. So you can see really rich staining of the collagen within even following the culture. And then within the synovium, so synovium contains 2 main cell types. There's fibroblast-like cells, which we're marking up here with Vimentin, and then macrophage-like cells, which we're labeling with CD68. So I did actually think the CD68 cells, the macrophages, would die off, but they're not. They're still there. So these cells are still quite viable and happy within the synovial cultures. Interleukin-1, just like in disc degeneration, is also involved in osteoarthritis. And one of the treatments we're looking at is trying to target interleukin-1. So it's really important that we knew whether this changed during culture. And what you can see is from immediate fixation to 21 days, there's no change in any of the culture systems between the culture and immediate fixation, which is excellent. Same for its receptor, because we need to know that the receptor is going to be the same, so it's not increasing responsiveness within our cultures. So what we're now doing is using this model to actually investigate, again, that gene therapy approach I talked about before within the disc, because this is actually in clinical trials at the moment within America, but they've got some interesting results in terms of potential effects with other drugs, which they can't unpick within patients. So because we can effectively grow 10 mini knees in the lab, all from one patient, we can actually look from one patient and treat with different co-treatments to actually see, how they're affected. So we're also looking at kind of minimally effective doses and everything. So this is all work that's kind of just started really. So I've not got any data to tell you about at the moment, but maybe I'll come back in a couple of years and tell you about this. But basically we're looking at what effects there is on kind of the gene expression, the proteins that they're producing, and also the secretory factors, and also really understanding, is it the synovium, the bone, or the cartilage that's taking up the gene therapy approach and actually delivering the IL-1RA to the knee model. In addition, we're also using the same model to look at the senolytics within osteoarthritis as well, because senescence is also increased in osteoarthritis. So just like with the disc, same kind of strategies, different senolytics, and then also the STING inhibitors. So this is just some initial data, again, looking at metabolic activity, and you can see that the high-grade osteoarthritis samples 1 or more variable, but do seem to get a greater effect with the ovanillin than the low-grade. And this is probably, we're hoping, because there's more senescent cells within the high-grade OA samples than the low-grade. But as I say, this is early doors, and we'll see more on this. So I'll stop there. Thank all the collaborators, PhD students, postdocs that have contributed to this work. It really is lots of different collaborators across the board, and all the funders that have supported the research. And I'll stop sharing so I can see people to answer questions.
Thank you very much, Professor Lemaitre, for that really wonderful presentation. It's really translationally motivated and from the basic science. So there are a lot of questions in the chat. So
why is Staph aureus more common? Yeah, so the first question I can see is why is Staph aureus more common in females? Maybe due to hormonal differences or disc circulation flow. So to be honest, we really don't know. This is a fairly new finding in the last sort of year or so, and something we want to look at more. It's not known why Staph aureus is more in the females and the discs, and we need to look within, other tissues as well. Is that the case elsewhere? But it is an interesting finding that we do want to look at more. Yeah.
So the next question is by Mashid. Based on the central disc circulation, how long would it take for the injectable material to get dissolved? Is it?
Yeah, so the gel system that we're injecting isn't biodegradable. So it's actually non-degradable. We've done kind of 18-month degradation studies and there's no evidence of any degradation at all. It's a synthetic gel, not a natural gel, but it's 90% water. So the idea is it's kind of an entanglement of polymer chains. So the way that it works is you get this kind of entanglement, so it's not a chemical cross-linking. 90% water, and then cells can migrate in, kind of push the polymer chains out of the way, and then they lay down the matrix within. So effectively, they turn the gel into the disc tissue with their own matrix that's being produced. So it's non-degradable, no issues of degradation or release of factors elsewhere, and it just turns into disc tissue.
So you basically addressed Muskan's question as well, who asked if the hydrogel used is biodegradable. So Tariq is asking, how are the stromal cells seeded in the gels? Differentiating, is it the mechanical or chemical properties of the gel that drive the differentiation of the stroma to the respective lineages? Are the gels biochemically functionalized, activated before or during cell culture?
Yeah, so interestingly, it's the gel alone that drives the differentiation. So we don't add any growth factors in. We don't need to kind of biochemically alter the gel system. We think it's because, so two reasons. One, it matches the mechanical properties really closely of the native disc. So it's kind of providing that mechanical stimuli that the cells should have within the disc tissue. And also it matches the osmolarity as well. So kind of the mechanical stiffness and also the osmolarity, we think that's why it's driving differentiation. There's no need for growth factors or any fancy functionalization. It's actually really cheap, the gel.
Yeah, so another question from Muskan. As degenerated discs have lower pH, altered nutrient flow, and mechanical stress, how does the hydrogel handle these harsh conditions?
Yeah, so it really doesn't mind. So we've also done work, I didn't have time today to talk about it. We've looked within media that mimics the kind of degenerate conditions. So low pH, low glucose, altered osmolarity. What's the other one that we do? Oh, on oxygen, and culture the cells within that environment with our gel. And again, they still differentiate, they still produce matrix. The gel seems to protect them. The gel is slightly alkaline. So we think it actually acts as a bit of a buffer to some of that, the kind of acidic pHs that you have within the disc as well.
Okay, the next question is from Mashid. Could we use mesenchymal cell enriched folded scaffolds instead of hydrogels as well, considering the narrow space inside the disc?
So I'm not sure what you mean by a folded scaffold. The idea of the hydrogel is that it injects in. So it's liquid when it injects in, flows into all the cracks and fissures, and then it gels. So we're not injecting in kind of a fully formed jelly, if you like. It's actually molding itself into the actual disc tissue.
And Mohan, stroma cells play a crucial role in intervertebral disc regeneration. What specific signaling pathways or microenvironmental factors are necessary to activate and sustain their regenerative potential?
So interestingly, we've actually found that with or without cells, the gel seems to induce regeneration. But the original idea was that we would need the cells, and we kind of thought that we'd need to replace those cells. Within the disc itself, you do have growth factors, but in a degenerate disc, you've got kind of that increase in cytokines and things as well. So it's kind of getting that balance, and that's why we think the gel will protect those stem cells. In the clinical trials that have happened so far, where they've been injecting mesenchymal stromal cells into the disc, there's no patient benefit being demonstrated. And I personally think that they're just dying when they're injecting them in because they're not being protected. And also they injected far too many cells, but yeah, we won't go there. I don't know whether that answers your question.
Okay. I think for the next question, I'll ask the people asking to ask them themselves. Muskan.
Yes. Sorry. Yes, thank you. So I wanted to know, is the gel like functionalized with any ligands or peptide before or during cell culture to like enhance cell adhesion, proliferation, or differentiation? They have same phenotype, I know.
Yeah, so no, we don't need to functionalize the gel at all. So the gel, it's based on, so it's a polymer chain, but then it also has clay nanoparticles in it. And the clay nanoparticles basically act like a bit like a sponge to any growth factors and also extracellular matrix components. So we think what's happening is when you culture it, any of the ECM that the cells are producing or the growth factors the cells produce, they get stuck in the actual gel. So that then kind of, that's why we then get that attraction of the other cells migrating into it. But yeah, we don't have to functionalize.
Okay, that's great. Yeah, it's fascinating. Thank you.
And actually, when we did try and functionalize it with HA, the cells didn't differentiate as well because I think they already thought that they had the right matrix there. So yeah, the simple system seems to work better.
Faji.
Hi, Christina. That was a very nice presentation. Thank you. I have a question regarding the bacteria. So is there any relationship for the microbiome with the OA progression compared to what you've seen in disc degeneration?
So that hasn't been looked at yet. We do want to look at that, and we have got quite a good bank of OA samples. It's just a matter of getting somebody to look at it. But yeah, that is something that we want to look at because the cartilage joint was always thought to be sterile, but so was the disc. So the disc was always thought to be a sterile environment. So we do now want to look, well, are there actually bacteria within the cartilage as well? You know, so that, yeah, that is something that's in the planning once I get someone to actually do it.
Thank you.
Mohan.
Oh, yes. Thank you so much, Professor, for the great presentation. I just have two questions back to back. So when you actually talked about the crosstalk, right, between the osteoarthritic changes and the osteochondral explants, I was thinking that maybe could other therapies like the platelets-rich plasma, could provide some sort of synergistic effect to it?
So platelet-rich plasma, it has been looked at as a potential, and it's mainly because of the growth factors which are present. The only trouble with platelet-rich plasma is it also contains lots of other things that can actually activate catabolic factors as well within the joint. So I personally don't think platelet-rich plasma is probably the best way to go, but it needs more investigation. So if anyone in the room's looking at it, I'm not insulting you.
Another question that I had was that since we talked about osteoarthritis and osteochondral explants, I was wondering like if there's any known impacts of systemic inflammation in patients with rheumatoid arthritis on disc degeneration.
So I'm not sure of the association with rheumatoid arthritis and disc degeneration. Actually, I'm not sure. So with osteoarthritis, you don't tend to get the systemic inflammation, but with rheumatoid arthritis, you do. I would imagine there's an increased incidence of disc degeneration in RA, but I don't think anyone's actually looked at it. I'm not sure. I'll have to look. Yeah, good question.
Thank you so much.
Fatma. Fatima, you want to ask the question? Why was the SHIP model chosen for evaluating NT-gel and NT-gel plus MEPC treatments?
Yeah, so the main purpose of the sheep model was a safety study. But we did also want to look at some efficacy outcomes as well, because obviously when you're doing a study on animals, you want to get as many questions as possible. Within disc studies, you need to look in larger animals if you're looking at regeneration. So small animals like mice and rats and rabbits, even pigs, they all contain notochordal cells, so they regenerate themselves quite easily. So we wanted a large animal model, and then sheep and goats are the best models for kind of looking at disc degeneration and regeneration. Sheep was the one that the partner on the grant had the ethics for, so that's why it's sheep. We are now looking at a goat model at the moment. The advantage of goat over sheep is they're a bit bigger, the discs, so you can get a little bit more of the gel system in there. So that's the kind of way forward that we're looking at the moment.
So yeah, so really wonderful work. So as you were talking, you said, given that you are talking about C. acnes, and given that C. acnes is commonly found in healthy skin, and as you showed, even in bovine disc. So do you think its presence in the disc represents true infection, or is it just normal part? Are you coming to the concept of disc microbiome?
Yes. So I think that the C. acnes, at least certain phylotypes, are kind of a normal microbiome within the disc. So we did see it within every single disc that we looked at, and all of the bovine discs that we've looked at, and also sheep as well, actually, we've looked at as well. So it is present in kind of every disc we look at. What I do think that we need to look at now is there differences in the strains. So with C. acnes, for example, most people have C. acnes as normal flora, but when you get acne, it's actually a different phylotype of the bacteria that comes up, and it actually kind of takes over, expresses different virulence factors, and that it kind of induces the pathogenesis. So what we want to look at now, if the funders will give us the money, which they said no, but anyway, I'll ask again. Is there different specific phylotypes? Are there particular virulence factors that are associated with disc degeneration? Or is it just normal microbiome and it's actually there to protect and things as well? So yeah, that's what we want to look at next.
Right. So the other question was, you see a correlation between C. acnes and Modic type 1 changes.
Yes.
So do you believe this is a direct cause-effect relationship, or could bacterial presence, could it be like a secondary consequence of an degenerating disc?
Yeah, so it could be either way. So it could be that the modic change, that you've got that because, and you've got kind of the fissures within the end plate, you've got end plate damage, and that's then causing more bacteria to come in or out of the disc and then causing those different amounts. In the animal studies where they've injected C. acne in, they do induce modic change, but the levels they've injected are far too high. So it's nothing like the levels that you see actually in human discs. So really we need to look, at the moment we don't have the evidence that says yes or no either way. And I think it could be cause or effect at the moment. We need more studies.
All right. Thank you very much. Really wonderful. I know that there are a lot of people with a lot of questions.
Yeah, I've never had that many questions. Thank you very much.
I'm sure Mike will kill me if I stop.
Yeah. Yeah, Christine, I have to say by far your gel is like the most advanced I've seen. Like, very promising for treatment. So, it's just a few questions on the so related to the bacteria. So I'm assuming you think, follow up on Faxon's question. So these are bacteria that are living within the disc cells. So these are dormant bacteria that you see, and I guess in pathological state or with the modic changes, you see different bacteria within the disc cell. So we're looking specifically at NP. You also see an AF, or is it just NP?
Yeah, so we see, we do see C. acnes in the NP and the AF. We see more in the NP though than we see in the AF, particularly, and you don't really see it in the outer AF. So you kind of get and increase as you go deeper in, which we think is because, so C. acnes is an anaerobe, it doesn't like high oxygen. So you see higher levels in the regions which is lower oxygen, basically. So the regions where it's just higher, you don't see as much. In terms of kind of which bacteria, so we've only looked so far at C. acnes and Staph aureus that we see both internalized. We do want to look at all the other bacteria that the kind of 16S sequencing have identified as potentially present in the disc to see whether they actually are, and not, again, whereabouts they are within the cells or they internalize. The other thing we don't know for sure is, are those bacteria still alive? Now, we think they probably are because we see them in so many cells. And of course, if the cell had killed it, it would be degrading it, it disappeared quite quickly. So the fact we see it in so many cells, we do think those bacteria are still alive. Whether they're active or whether they're dormant, again, we don't know. We need to look at it in more detail, really. Yeah, it's opened up so many more questions. I kind of thought doing that study that we wouldn't find them and it would just rule it out, but actually it's opened up way more questions.
Exactly. And the other one is on a little bit follow up on Muskan's question, so related to the environment, the degenerative environment of the disc. So given what we know so far with calcification of end plate, the fissures you mentioned, the tissue being more fibrotic, of course, dehydrated. So I understand that when you inject your gel, so it provides some sort of level of rehydration. You could also put cells in there, so the cells may thrive in those areas where the gel is present, I would imagine.
Yep.
So given, and you have seen, of course, in your in vivo models and ex vivo models, increase in matrix proteins. But in the case of, let's say, a patient, so how do you foresee any issues? I would imagine variability and so on when you inject this in an actual patient. And how long do you think it'll take for, let's say, a regeneration to take place? And finally, have you looked at like markers of pain and so on in these models to see if there's any changes with the gel? And in case of symptomatic relief, what do you know?
Yeah, so lots of questions there. So if I don't answer one, so I guess, so in terms of the effects of calcium and the kind of degenerate environment, so we have looked at media with increased calcium as well. And actually calcium is the one thing we can't inhibit with the gel. So we do probably think that we need to kind of look carefully at patients, and those which have end plate damage and really bad end plates or calcified end plates are probably ones that you wouldn't target. But also those patients with really calcified end plates would also have reduced nutrient supply to the disc, so probably are not the best for regeneration. So you kind of want to look at the ones which have still got non-calcified end plates for targeting. In terms of the pain markers, and then you'll have to repeat the others because I can't remember what the other ones were. So we have looked at kind of nerve growth factor, vascular endothelial growth factor, because obviously often you get kind of the vasculature growing in as well as the nerves. We do see a decrease in both vascular endothelial growth factor and nerve growth factor, both within the goat model and the human cadaveric study. So we saw that you do get that decrease in those factors within those treatments. We didn't look at anything like NT3 or anything like that in terms of other factors. With the sheep model, it was a kind of block design, so we couldn't do kind of pain measures on the sheep, and it was only 2 sheep in each group. But that is something that they're doing at the moment. Well, planned for the large animal goat study is the plan is to put Fitbit collars on the goats and then actually be able to monitor kind of pain responses and movement and things to actually see are we targeting the pain responses and things as well. I'm sorry, what was the other? There was something else in the middle.
It's more, no, you're answering very well. So it's the time frame for regeneration, especially in the patients. How much matrix protein or tissue do you expect is required, or do you have to re-inject again, just to push that barrier up enough to get enough matrix to actually see some sort of symptomatic relief?
Yeah, so we're hoping that you can do it with a single injection. So because the gel kind of mimics the mechanics of the disc straight away, and because it fills all the cracks and fissures, we could do a single injection to kind of provide that initial mechanical response. Within the organ culture study with the human discs, even just after 4 weeks, the hydrogel had started to turn into disc matrix. So where it was all kind of the thin cracks and fissures, they'd all got matrix in them. Where it was a big lump in the middle, it was filling. So obviously, you wouldn't expect it to do it in 4 weeks. In the sheep, the whole region with the gel has turned into matrix, and actually it's really hard to identify what's gel and what's not. That's after 3 months. So I would say probably in a human, you'd expect it to be probably a little bit slower than a sheep. So I would say probably in 6 months that hopefully we would actually kind of have regenerated the tissue. I was originally thinking it would take a lot longer, but seeing how fast the 4-week culture was in the organ culture, actually, I think it's much faster than we thought it would be.
Oh, perfect. It's just to get an idea or a sense of your timeline. If you're going to do clinical trial, what are you, when are you going to measure? What's your endpoint? How long are you going to wait? So on. Yeah, that's just
so
trying to.
So for the clinical trial, they'd be looking at kind of a 2-year follow-up, which they have to do anyway for safety elements as well. The clinicians that we've talked to, and I'm not sure what the company is going for directly, but they've talked about having kind of initial treatment where a patient's got a discectomy on one level, the next level up is starting to herniate, or it's degenerate but not herniating yet, that they treat that disc, because then the patient's going in for surgery anyway, and the main output measure would be MRI signal intensity and things like that, and movement, recovery and things. That would be the first, but then after that, obviously it would be different stages of degeneration, or can you refill after a herniation is something we're looking at as well, to see whether, depending on how much damage there is, how much can you actually inject in and keep it in there.
So in the interest of time, I think let's have Derek as the last question. Short and sweet, Derek.
Yeah, as always, Christine, nice talk. I was curious and interested in your ex vivo culture model for looking at the senescence in the sample. So I was just interested in the model, and I recall seeing those in the past where you have sort of a silicon disc that keeps them from swelling outwards. Do you also control for swelling up and out of that silicon ring? What's holding it down? And then second, how much does the tissue swelling and mechanics there, or osmolarity, influence the stress-induced senescence?
So in terms of the osmolarity, I'll answer that bit first. So we actually control the osmolarity in the media. So we culture them in a media that mimics the kind of degenerate environment. So the osmolarity is actually very similar to the disc tissue. So with human discs, we don't see that much swelling at all upwards. If you just constrain it kind of circumferentially around it, stop using big words. If you constrain it around it, it doesn't actually swell up and out. If you grow bovine tissue in it, it does swell above, but only over the first couple of days. And then basically what you do is you cheat and slice the top off, and the rest of it's fine. But yeah, human disc, it doesn't seem to swell over. And the reason why we're using kind of the tissue cultures for the senescence is then we've not got any of the issues of, trying to isolate the cells, and the senescent cells don't stick down properly. You've not got the issues of, growing them out and inducing senescence as well. It's actually then as close as possible to kind of the native tissue, and we can use surgical samples. So the size of the kind of the perspex rings, or we switch to silicone rings now, the size of them is designed that most larger herniation tissue, we can actually get kind of at least 6 samples from that surgical sample. So we can actually make use of patient surgical waste material and get lots of different tissues that we can utilize.
And I didn't make the connection, but were you also doing that with the osteochondral plugs? I don't recall if you're looking at
so with the osteochondral, it's the osteochondral plug together with the synovium culture. So we're kind of doing it as the mini knee cultures, we're calling it.
Very cool. Thank you.
Thank you very much, Christine, for that wonderful presentation. I think we have 2 students. Who's going to moderate? Yeah, Kyle. Tariq, just show yours.
Okay. So good afternoon, everyone. My name is Tariq Leilat. I'm a PhD candidate in the lab of Dr. Rahul Gowri and co-supervised by Dr. Derek Rosenzweig. And I'll be the moderator for today's student presentations. Just a quick reminder before I start introducing the first student that there will be feedback forms that will appear after each presentation for you to give feedback on the students' presentations. So the first student for today's presentations is Cassandra De La Roca, and it is my pleasure to introduce her. Cassandra completed her Bachelor of Science in Anatomy and Cell Biology at McGill University and is currently pursuing a Master's in Surgical and Interventional Sciences with a concentration in surgical outcomes research. She conducts her research in the lab of Dr. Andrew Zakari at the Department of Obstetrics and Gynecology at the Royal Victoria Hospital. And her work is centered on improving patient recovery by challenging conventional prescribing practices. So today she will be presenting her research titled Eliminating Narcotic Prescriptions from Outpatient Minimally Invasive Gynecologic Surgery, a Randomized Controlled Trial. Cassandra, the floor is yours.
Thank you, Tarek. I've already shared my screen. Can everyone see it?
Yes.
Perfect. So I'll hop right in. First off, I have no conflict of interest to disclose. So can you recall a time when you or a loved one had a minor surgery and you left the hospital with an opioid prescription? Did you take those opioid tablets, or are they collecting dust in your medicine cabinet? So when opioids become more easily accessible in our community, they can become a source for recreational use and abuse, and even addiction or use of stronger illicit narcotics. They can lead to serious accidents and fatalities, especially if they get into the wrong hands. And even when used appropriately, opioid use does come with a significant risk of adverse effects, such as nausea and vomiting and constipation, which may negatively affect the post-op recovery. So over the last several decades, misuse and abuse of opioids has become a major public health crisis, especially since the COVID-19 pandemic. Today, 1 out of every 22 deaths in North America is now attributable to unintentional opioid toxicity, and nearly 1 in 3 of these deaths are related to opioids obtained through medical prescription. So what can be done? Can opioid exposure be avoided in certain cases? This is where minimally invasive surgery comes in. So minimally invasive gynecologic surgery, or MIGS for short, are surgeries that only require a few small incisions, since a camera called a laparoscope is used to operate. These laparoscopic surgeries allow patients to go home the same day. They're associated with less pain and opioid use and a shorter recovery period. But although surgical techniques are evolving, there's still no guidelines on appropriate opioid prescribing in gynecology. A wide range of prescribing patterns are being reported by gynecologists, even though studies show that patients often use less than half of their prescribed opioids following MIGS. There you go. And with an up to 7% chance of addiction in opioid-naive patients, that's 1 in 14, reckless prescribing patterns puts this patient population at risk. So although the literature has emphasized the importance of taking action to reduce opioid prescribing, to date, no RCT has evaluated the complete elimination of opioids post-operatively in MIGS. So we aimed to answer the question. For select patients discharged the same day after MIGS, would a multimodal opioid-free prescription for analgesia result in non-inferior post-operative pain compared to an opioid-containing prescription? So we conducted a single-blind, randomized controlled trial on patients undergoing elective MIGS with same-day discharge. The trial essentially compared 2 groups of patients who were sent home with different prescriptions for pain relief after surgery. Any patients with an allergy to the study medications or a chronic pain condition or who regularly used analgesia were excluded from participation. In order to minimize variability between cases, the OR staff adhered to a standardized anesthesia protocol and also a surgical checklist for all patients. So, for example, the incision size and number were standardized for all patients. And any deviations from these protocols resulted in exclusion from randomization. So how it worked, upon completion of their surgery,
participants were randomized to one of two sets of discharge prescriptions for multimodal analgesia, or pain relief. So one group on the left went home with only non-opioid pain relief medication, so in this case, acetaminophen and naproxen, while the other group went home with those same medications, but also a few opioid tablets, in this case, hydromorphone. And don't worry, participants in the opioid-free group did have access to an emergency number that they could call to reach a physician in case they needed a rescue prescription. To collect data, a researcher blinded to the allocation, called patients 1 day and 1 week after their surgery to ask them questions about their pain control and recovery experience. And our primary outcome was participant-reported pain score on post-op day 1 using an 11-point numerical rating scale, pictured right here. Our secondary outcomes were other patient-reported recovery parameters, such as pain score on post-op day 7, opioid consumption, mobility, and satisfaction. So our sample size was powered to detect a difference of 1 point on this 11-point numeric rating scale, since pain studies have shown this to be the minimal clinically important difference. And since this study was designed as a non-inferiority study, pain control would be considered non-inferior in the opioid-free group if the mean pain score fell within 1 point of the restrictive opioid group's pain score. So more analysis. The baseline characteristics were descriptively analyzed between groups. To compare the post-op data between the two groups, the Wilcoxon rank sum non-parametric test was used for continuous variables, and Fisher exact test or chi-square test for categorical variables, and two-sided p-value of less than 0.05 was considered statistically significant, as usual. Generalized linear regression analysis were also applied and performed for all independent variables to search for predictive factors contributing to post-op pain scores. And lastly, we also performed an exploratory subgroup analysis of pain scores among different types of surgeries. So from the 521 screened minimally invasive gynecologic surgeries, 206 patients were eligible, 137 were recruited, 110 were randomized, and then 103 completed the study. Here's a list of demographics and surgical characteristics that we collected. I won't go through all of them, but what's important is that they were balanced between the two groups. And now the exciting part. So we found that there was no significant difference in mean pain score on post-operative day 1 between both groups. Similarly, there was no significant difference in pain scores on post-operative day 7 either. Regarding secondary outcomes, interestingly, only 27.8% of patients in the restrictive opioid prescription group reported using any opioids by post-operative day 1. And then only 11% reported subsequent opioid use by post-operative day 7. Remarkably, no patients in the opioid-free arm required a rescue prescription for opioids. On post-operative day 1, nearly all patients in both groups reported their pain to be overall well-controlled. And also on post-operative day 1, nearly all patients in both groups stated that their pain control was adequate enough for them to be mobile and perform basic tasks of living. The linear regression analysis identified no significant predictive factors associated with pain, except for younger age being associated with higher reported mean pain scores. Interestingly, BMI, prior surgery, procedure length, estimated blood loss, and mini laparotomy all were not predictive factors of post-operative pain, even though we thought they might be. Subgroup analyses were done for patients undergoing total laparoscopic hysterectomy and myomectomy, as those were the most prominent subgroups of surgeries, but the results for these groups were consistent with overall findings. So to recap, in this RCT, eliminating opioids from exit prescriptions following outpatient MIGS did not result in inferior pain control compared to an opioid-containing exit prescription. No patients in the opioid-free group required rescue analgesia, and both groups had similar satisfaction, mobility, and other post-op outcomes of interest. So these findings are in keeping with the literature, which has consistently shown not only low pain scores following MIGS, but also frequent opioid overprescribing by providers. So one such study seen here was a non-inferiority RCT done by Yuan and et al. That compared a standard opioid prescription to a restrictive one where opioids were only prescribed upon patient request. And this study found that only 29% of patients in the restrictive opioid prescription consumed opioids within the first week, which was similar to our findings of 28% in the restrictive opioid group. Their study also found that patient satisfaction was non-inferior in the restrictive group, despite that group consuming significantly fewer opioids. So similar studies have been replicated in the gynecology oncology MIGS literature that have showed that implementing a restrictive opioid prescribing protocol did not adversely affect patient reported pain scores or satisfaction, while significantly reducing opioid consumption compared to past cohorts. So to conclude, the findings of this study showed us that opioids can be safely omitted in this patient population. Our research highlights the importance of opioid stewardship by providers, which is an important step in addressing the nationwide opioid crisis. We hope that the findings of our study can contribute to quality improvement initiatives targeting patients and provider education on strategies to reduce or eliminate opioids after MIGS. And lastly, the only thing to be explored is determining an ideal anesthesia regimen to optimize post-op pain and recovery. So thank you. Oh, I'd like to acknowledge my team, especially the MUHC Royal Victoria Hospital Obstetrics and Gynae Department, who I've worked closely with, as well as gynecology oncologists, anesthesia, and the whole nursing team who helped us employ our protocols during surgeries. And yeah, that's it. So thank you. Any questions? Do we have time for questions?
We have maybe a couple minutes for questions, and there's
Okay.
already a question in the chat. I don't know if you can see it, or if not, maybe Mohan can.
Yeah, I can read. I can see
perfect.
it. Okay, one question. In your study, did patients receive any preoperative drinks or nutritional support to optimize their preparation for the procedure, similar to the strategies used in ARIS protocols? That's a great question. We did not implement any ARIS protocols, but I actually took a class on this, but this was after our protocol had been written and we had started the study. But I definitely do think that that implementing ARIS protocols has the potential to reduce post-operative pain. It's definitely a study that can be done in the future. Yeah. And should I read the next one in the chat?
Yes.
Have these results already led to a change in prescription policies at your center? So no changes have been made yet, but we're hoping by diffusing our results and presenting in conferences, both like local within the hospital and also just national and international, that hopefully policies can be changed and guidelines can be written and officially created. That's the end goal of all of this.
Maybe we can take one last question before moving to the next talk.
Yeah, sure. I could ask a question. Great talk, actually, very interesting. And yeah, I think there's a lot of promise there for the switch from NSAIDs, from opiates to NSAIDs. But have you looked at like any changes, let's say, in gastrointestinal bleeding or patients complaining about gastro irritation? Like, do you foresee that as being a problem when switching direct or just exclusively to NSAIDs over opioids?
So yeah, that actually was one of the groups of patients that we ended up excluding from participation due to like not being able to really consume NSAIDs were people with like GI issues, like whose doctors recommended that they don't take any. But for certain patients, their doctor said it would be fine to take them, like if they're only taking these medications for a short term, like such as a week or even less. Like usually it won't make too much of a difference for them. But at that point, I think it comes down to what their doctor recommends. But in terms of the population that our results can apply to, it's the patient population who do not have chronic pain, who can take NSAIDs, like that's a good point.
Thank you so much, Cassandra. And I think for the interest of time, we'll move to the next speaker.
Yeah, thank you so much.
So it is my pleasure now to introduce our next speaker, Thanasis Babouras. Thanasis holds a Bachelor of Computer Science from Concordia University, which he completed in 2019. And his interest in the medical field was sparked by his experience with his family's rehabilitation clinic in Greece, leading him to focus his research on leveraging computer vision and machine learning to enhance health outcomes. Under the supervision of Dr. Paul Martineau and Dr. Thomas Fevens, Thanasis is exploring innovative approaches to real-time injury risk assessment in orthopedics. And today he will be presenting his work titled Accessible Real-Time ACL Injury Risk Assessment Using Pose Estimation and Deep Learning. Thank you.
All right.
The floor is yours, Thanasis.
Yeah, thank you, Tarek, for the introduction. So very happy to be presenting some of our research today. So I can get right into it. So the goal of our research basically is to utilize modern machine learning techniques in order to create a tool for accurate anterior cruciate ligament risk analysis. And our plan to do that is to develop a portable, low-cost application in order to record a person performing an athletic stress test and analyze their biomechanics throughout the exercise. So just to get a bit of a background on ACL injuries. So the ACL tears account for about more than 50% of the knee injuries. And there are approximately 400,000 ACL reconstructions every year in the US alone, with a huge system cost. And up to 80% of those are a result of a non-contact injury. So lower limb biomechanics have been associated with poor neuromuscular control, and they are a known risk factor for ACL injury, and thus a target for intervention. So the coronal angle and the sagittal angle when someone's performing certain athletic stress tests have been connected to high risk of injury. Typically, to perform this sort of analysis, to look at the angles and see if a person's at high risk, you need a motion analysis laboratory, which uses multiple cameras. It's high cost. It's just on a single location and typically uses cumbersome markers. There are some solutions now that are markerless, but typically that's how the analysis is performed. And one of the main jumps that has been studied in the past is the drop vertical jump, which is a validated tool for assessing lower limb biomechanics and the risk of ACL injury. So they basically get on top of a box, drop off of the box, and then jump up as high as they can. And depending on their knee angles throughout that jump, we can predict if they're at risk of ACL injury. And so our research objective was to create a portable and inexpensive solution to perform real-time screening of ACL injury risk, as mentioned before. Now, initially, we developed a solution that used Microsoft's Kinect V2 camera for skeleton tracking in order to track a drop vertical jump or even other athletic stress tests. And using that solution, we initially validated against the gold standard of ICON system. And after that, we found good predictive ability of ACL injury on a cohort of 102 varsity athletes using the drop vertical jump. So just taking the peak knee flexion throughout a drop vertical jump from our system, we were able to predict ACL injury with 80% sensitivity and 74% specificity. Now, the issue we had with that is that the availability of the Kinect V2, as some of you might know, it's not available anymore and it's really hard to get. So our initial thought was to experiment with other depth sensors and see if we can get similar predictive ability. We first looked at the Azure Kinect and compared it to the Kinect V2, though we did find that it wasn't as good in predicting the ACL injury from a drop vertical jump. And additionally, the Azure Kinect has become also discontinued, same as the Kinect V2. We further looked at another sensor, the Z2i from a company called Stereolab. So that is also an expensive solution with costing more than $700, and it also requires a powerful graphics card. So what we decided to do moving forward is explore pose estimation techniques, which is pose estimation is a computer vision task to infer human pose from a 2D image. There are many solutions that are available, so a lot of them are open source as well, and we decided to work with Google's MediaPipe due to the ease of use and the availability. The only issue with those is that they haven't excessively been validated yet. So what we did in order to validate, we developed a smartphone application using the MediaPipe framework, where you can track a person's joints in real time, 3D joints in real time. And we published a comparison with the Kinect V2 system that we had using data from 254 varsity athletes. We looked at the drop vertical jump and 2 other athletic stress tests, the single leg hop and the single leg squat. And we found small differences, small coronal angle differences and moderate sagittal knee angle differences. And we also further found excellent agreement in the predictions compared to the ones given from our system using the Kinect V2. And we're currently working on also a comparison with the gold standard system. It's called the Qualisys, where we found that it performs similarly to the Kinect V2 when compared to the gold standard. Now, the results from our comparisons we presented in the American College of Sports Medicine annual meeting, but also the International Conference of Information Processing and Computer Assisted Interventions of 2024. Now, going further, we also started working on a better prediction model. So since we've gathered data from over 700 unique varsity athletes during the preseason physicals, our goal was to focus less on the singular values, the coronal and sagittal, but to look at the continuous data throughout a jump. And so the goal was to use that data to train a model to have better predictive ability. Initially, we looked at a support vector machine model to use the continuous angles and see if we can get a good prediction. Though the results were not great, one of the issues is that we had very low positive cases, so very few ACL injuries in our cohort. And in those athletes that we captured, there may be a lot of athletes that were at high risk that didn't actually injure their knee yet. So those were captured as false negatives and were affecting the results. As such, we decided to explore an unsupervised learning approach, which avoids the above issues. And looking into related work a bit, so we found other models that have been used for similar approaches, though not exactly the same. So the spatial temporal graph convolutional network has been used before for skeleton-based action recognition, which captures spatial and temporal patterns of movement, though it needs labeled data to work. And also autoencoder models have been used for anomaly detection before, though they're used to learn features of unlabeled data that they haven't been used in this approach before. So our idea was to combine the 2 architectures in a single model. And for that, we developed a deep learning model that does combine the 2 architectures. And we used the joint positions of a vertical jump directly, so the XYZ positions across a number of frames. And we kept the lower body and spine joints, 9 total joints, and added the center of gravity in order to have a good estimate of the upper body as well. The reason we did that is because having the hands in the prediction sort of introduced a lot of noise from the pose estimation. And finally, we used the movement just up to initial contact, which had the best predictive ability, which is interesting since it shows that there might be something in the way they load before performing the jump that puts them at high risk. And using a data set of 561 drop vertical jump recordings, we were able to predict high risk jumps with a 92% sensitivity and 72% specificity. And we submitted a paper with a model we developed to the International Conference of Machine Learning on Machine Learning. So to give a brief overview of all our research so far, we have validated our smartphone solution compared to the existing solution that we had. We validated its tracking ability compared to the gold standard, and we'll be publishing that soon. And we've also developed a deep learning ACL injury prediction model for the drop vertical jump. Now, for our next steps, we're looking to, since deep learning models are a bit of a black box, we're looking to have a better interpretation of the model results. We want to also explore other athletic stress tests and their predictive ability, the single leg hop and the single leg squat. And finally, we're looking to release the application we're building to the public so they can start doing these assessments by themselves and collect the data from that to train a more accurate model. And that's everything. Thank you very much.
Thank you. And I think we already have a question. Yeah, a couple questions.
Let me see. So I think the first one, do you use one or two phones? If Anton, do you think using two phones will improve the result? Why not looking at the transverse plane? So yeah, thank you very much for that question. So we are using just one phone. The idea was to not have the requirement of having two phones. That improves the result usually due to having stereo vision, basically, so you can get better 3D positions. But the idea was to have it as simple as possible. So we're just using the one phone, and the transverse plane we're not focusing as much on because solutions such as the media pipe solution, which is from pose estimation, doesn't calculate the transverse rotations as well compared to, let's say, a Vicon system, a full marker system. So we're just looking at the coronal and sagittal angles, which seem to be more accurate. And if that answers your question. I can answer the second one. In addition to pose estimation, have you considered integrated biosensors to measure muscle activity, from hamstrings or quadriceps during dynamic movements to improve the accuracy of ACL injury predictions? So yeah, so far we haven't looked into that, integrating some additional sensor to get extra information, but that would definitely be an interesting way to move forward, maybe adding some inertial sensor as well to get more accurate results. But having something to measure muscle activity would be definitely interesting as well. Yeah.
So, yeah.
One more question. Yes.
Yeah.
Forgive me for my ignorance on this, but are there sex-related differences between this assessment?
So yeah, in terms of ACL injury, this is something I didn't mention. Females are typically thought to have a higher risk of injury for ACL injury, sometimes up to 7 times the risk. Now, this is something that in our data set we haven't noticed. So most of the injuries actually we've seen, because we've seen athletes from McGill and Concordia, most of the injuries have been males. So this is not something we've seen in our experience, but it is there in the literature that they're much higher at risk.
So in the future, will you need to include this in your assessment risk?
Yeah, this is something we're looking to do as well, to create a, let's say, multimodal model where it takes the biomechanics assessment, but can also add demographic data in the prediction, and it can perhaps improve the overall result.
So in the interest of time, I think I would like to thank all our speakers, Professor Christine Lemaitre, Cassandra De La Roca, and Thanasis Pavouras for wonderful presentations. And thank you very much. And I'd like to thank Tariq Khailat for moderating. And thank you everyone for attending the meeting, and have a wonderful spring. It's getting better.
Yeah.
Hi everyone. Thank you very much.
Bye. Thank you.
Bye-bye.
Bye.